Good morning, one and all. Today we will continue now the discussion of the tools of biotechnology. So, under the tools of biotechnology or recombinant DNA technology, we have already discussed about in details restriction endonuclease enzyme. Then we spoke about agar-rose gel electrophoresis. Now, we also started talking about the cloning vectors. So what we will do is we will restart our discussion from cloning vectors. We'll continue with some revision of what we have learned last class, and then we'll continue with the discussion of what are these cloning factors and some important points. Now, you see in the cloning vector, they are basically nothing but they are kind of carriers which can carry the foreign gene into the host cell, because we have understood that this is basically a process wherein a foreign gene is often inserted into a cell which does not belong to its own, therefore you call it as a foreign gene. It may belong to a different species entirely, or it might be a different subspecies. So that is why the host cell may not accept and allow the expression of this cloning vector or the functioning of this, expression of the foreign gene or the functioning of the gene. Therefore, we use a vector whose literal meaning is a carrier who can easily carry the foreign gene into the host and stabilize that gene inside the host for its proper functioning. Therefore, in that case, it is easy to understand that these cloning vectors must be something that the host can easily recognize. That is why, based upon the different hosts, we have different types of cloning vectors. The most commonly used host when we talk about expression of certain gene to get certain products out of it is bacteria. And for a bacteria to be used as a host, the most commonly used cloning vector is a plasmid gene. Now we know that plasmid gene is basically a non-chromosomal bacterial DNA only, right? And it will usually confer certain additional spatial advantages or properties to the bacteria. So when we isolate a plasmid vector from a bacteria and use that vector as a carrier for our gene of interest, then the host can easily take it up, because as usual the plasmid vector is naturally a part of the host bacteria itself. Bacteriophages are also sometimes used based on the fact that the bacteriophages are the viruses which can infect the host, that is bacteria. And viruses, we know, have a property that it will, whenever it will infect, it will basically introduce the foreign DNA, its own DNA, into the host cell, right? So when we try to introduce our foreign gene with the viral DNA, then while introducing the viral DNA into the bacterial host, the foreign gene will also get introduced, correct? Now, bacteriophages, we do not have a detailed discussion as far as our syllabus is concerned. Our syllabus is mainly talking about the plasmid vectors as such and how we use them. So a plasmid to be used as a vector must have certain properties. The common properties which are necessary for a vector to be used for cloning purpose are: number one, origin of replication, then cloning sites, and the selectable markers. The origin of replication and cloning sites we have discussed earlier. Origin of replication is basically a part within the bacterial DNA. Okay, within bacterial DNA, a part of the DNA where from the origin of replication can take place. That is, the replication process can start. When you will learn about replication in DNA, you will understand that DNA does not undergo replication randomly from here and there. It always undergoes replication from a very specific point, and that point is usually called as origin of replication. That part of the sequence also has some speciality in terms of the nucleotide sequence configurations there. Then next one is the cloning site, are those sites where we see various types of recognition sequence of different restriction endonuclease enzyme. The most common cloning site that we talk about, and we also have in our syllabus, is cloning vector that we talk about is PBR322, right? Which was discovered by two scientists, Bolivar and Rodriguez. So it is known as PBR322, which is a cloning vector mainly for the host, which are E. coli. Okay, it is a cloning vector for E. coli host. Is the Escherichia coli bacteria. Right. Now, what we see here in this PBR322 cloning vector, there are various names here: PST1, PVU1, EcoR1, etc. These are basically nothing but the names of enzymes, that is restriction endonuclease enzyme. So why are these names written here? It basically means that at these particular regions, there are some specific recognition sequences, the sequences which can be recognized by these particular enzymes. Okay, that is why we call them as cloning sites, because cloning sites means the sites where you can clone or you can add the foreign gene of interest. Okay, so we have also learned in the previous class that cloning sites must be unique site. Unique site means each enzyme must have only one cloning site and not multiple, because if there are multiple cloning site for one particular enzyme, then our plasmid will break down into two pieces. But we want our plasmid to mainly open up, something like this. Here we see that you have a cloning vector plasmid, because EcoR1 is present at two locations. So what is happening is, if you treat it with the enzyme, then it is breaking down into two pieces here, and thus it becomes useless, right? However, if the cloning site is present at only one location, then it will open up where you can add your gene of interest. That is how the gene of interest can be added to the cloning site. But there has to be multiple cloning sites for different types of enzymes, because in that case you can use that particular vector for different types of enzymes, right? Now, these were the topics or discussion about the origin of replication and what we call as the cloning sites. Now, today we will be talking more about what we call as selectable marker. This is another property that must be there in a vector or in a plasmid to be used as a vector. Now, what is this selectable marker? Let us try to look at that. Okay. For understanding selectable marker, we look at it in this way, wherein we say that when the gene of interest is added to the plasmid, by the process of recombination, we get a molecule which is known as recombinant DNA. This is called as a recombinant DNA. Now, this recombinant DNA, when it is introduced into the host, the process that takes place is known as transformation. Okay. So, successful entry of that recombinant DNA inside the host. Now, the problem is, when we do this process, then we cannot actually see the process happening with our eyes, because these are DNA molecules we are talking about. Bacteria we are talking about. So this cannot be observed with naked eyes to identify whether it has happened successfully or not. So we need a process which is called a selection. Okay? We need a selection process so that you can select successful recombinant and transformant ones from the remaining. Because we have noted here, in one of the possibility, it is possible that neither the DNA got recombined with the vector, so unsuccessful recombination, nor there is any transformation. So both recombination and transformation are unsuccessful in the first case. In the second case, recombination has happened successfully, that is good, but the recombinant DNA did not enter the host, so it has not shown transformation process. In the third case, what has happened is recombination did not happen at the first place. Only the vector alone entered the host cell. So transformation did happen successfully, but recombination was unsuccessful. In the fourth case, recombination as well as transformation both have happened successfully. And this is the one which we want to select out. This cell, host cell, we want to select out from all the remaining types which might be present in our reaction mixture. Okay, so that is the technique we are going to use. Now for that process, how do you do the selection process? How do you select out that what has entered or what has not entered, or which one has successfully transformed and successfully recombined, etc. So for that, we look for the presence of some special type of genes in the plasmid vector. And these genes are called as marker genes. Since they help in the selection, so we also sometimes call it as selectable marker gene. Okay? Since they help it in selection, so we call it as a selectable marker gene. Now, commonly there are two types of marker genes which are used. One is called as antibiotic resistance gene, and the other is called as color coding method, or it's basically a beta galactosidase gene. Right? Let us first understand the antibiotic resistance gene. How does it work? Okay. Before we continue, does anyone has any question to clear here from whatever we have discussed about cloning vector till now? If anyone has anything to ask, please tell me. So let us say this is your plasmid vector. You can see that in your plasmid vector, the actual diagram which was shown, here a pink portion is shown. Okay, and here another pink portion, both are labeled as AMPR and PETR. Right? If you look at this diagram, it is clearly mentioned one of this gene, red colored this gene, is known as tetracycline resistance gene. Now understand what type of gene is it. Antibiotic resistance gene you must have studied in class 11 when you studied about plasmid. This is a type of plasmid which is called as R plasmid. R plasmid contains certain gene which provide resistance to the bacteria towards antibiotics. We know that the bacteria will not survive if we put it in some antibiotics. We know that antibiotics can kill bacteria. Now what happens is, if I am talking about a plasmid which has a gene which is known as tetracycline resistance gene. It is called as TETR. TETR stands for tetracycline resistance gene. It means that wherever this plasmid will be present in whatever type of host you introduce this particular plasmid, that host will become resistant to the tetracycline antibiotic. So now if this particular bacteria you put it in a tetracycline medium, then that medium, in that medium, the bacteria will be able to survive. Why? Because the bacteria contain a plasmid which contain a gene responsible for resistance to that particular antibiotic. Similarly, another gene that is thrown in the PBR322 is AMPR. This stands for another antibiotic known as ampicillin. This is ampicillin resistance gene. So similar to tetracycline resistant gene, if this particular plasmid is present inside any host, then that host bacteria will become resistant to ampicillin containing medium, or will become resistant to ampicillin antibiotic. Means, in simple words, ampicillin antibiotic or tetracycline antibiotic will not affect the bacteria, because the bacteria is containing a plasmid which contain a gene responsible for resistance to these antibiotics. Okay, please keep this very clear in your mind. Now, if you look at the diagram previous, you see in the ampicillin resistance gene, within that gene, there are two cloning sites, one recognized by PVO1 and the other is recognized by PST1. Similarly, within the tetracycline resistance gene, you have one cloning site recognized by BAMH1 and the other site recognized by SAM1. Okay, so we can understand what it means. If I am going to treat this particular plasmid, we normally call it as digestion. Digestion basically means breakdown. Okay, so when we use, let's say, BAMH1 enzyme, what will happen? That enzyme will go and cut the DNA within the tetracycline resistance gene. Okay, try to understand. So here we know that at this point you have an recognition sequence for the enzyme known as BAMH1. So if I put BAMH1, okay, then what will happen is the plasmid will open up in such a way that ampicillin resistant gene will be present there without any problem. But BAMH1 enzyme will cut this gene, tetracycline resistant gene, from here to here and from here to here into two pieces, right? Creating a gap in between. In this gap, if I want, I can put some foreign gene of interest. Now, this phenomena, understand properly, is known as insertional inactivation. What does it mean? When you have cut the tetracycline resistant gene in between and introduced some foreign gene within it, immediately what happened is your gene has become non-functional. Means that tetracycline resistance gene has become non-functional. It will not function anymore. Got it? So we say that the tetracycline resistance gene is inactivated. How? By insertion of a foreign gene within that gene. There was a insertional inactivation of the tetracycline resistant gene. Now this plasmid, if it enters a host cell, then will that host cell still be resistant to tetracycline? No. Earlier, the host cell containing the untreated plasmid, if this one you put it in the host cell, right, then the host will be, when you put it in a host will be resistant to ampicillin, which is an antibiotic, as well as tetracycline, which is another antibiotic. But if you put this one inside a host, then that host will become, it will remain resistant to ampicillin only, but it will become susceptible to tetracycline. Understood the point? Now let us understand how this process will be useful for selection. Okay. Now you see what will happen. Let us take a medium. This is a petri dish. In this petri dish, I will put ampicillin as an antibiotic. Here you have ampicillin containing bacterial growth media. So you put the bacteria here, the bacteria will grow. But here it contains ampicillin antibiotic. Now what is our aim? Out of the 4 possibilities here, I have to select the 4th possibility from rest of them. However, this vial contains all the probable 4 possibilities. So when I am going to pour this vial here, then all the 4 possible types of bacteria may be present here. But since this media contain ampicillin, so the first type of bacteria will not be able to grow. Why? because this bacteria does not have the plasmid inside, and therefore the bacteria does not contain any ampicillin resistance property. It will not be resistant to ampicillin because the bacteria does not contain the plasmid. Second one again will not be able to grow for the same reason, because although here recombination did happen, but transformation did not happen. So again the bacteria does not contain the plasmid, so it is not resistant to ampicillin. Okay. Therefore, we have successfully selected the transformant ones from the non-transformant ones. Means those cases where transformation did not happen, they have been discarded. Okay, and we have selected only the third and fourth ones where transformation did happen. Please understand, the first and second one did not grow because transformation did not happen. Therefore, the bacteria did not acquire the plasmid, and therefore the bacteria did not become resistant to the ampicillin. So the media which contain ampicillin will be responsible for the death of the bacteria. Now, coming to the third and fourth possibility. In the third one, there is unsuccessful recombination. Both has successful transformation, but the third one has unsuccessful recombination. Fourth one has successful recombination. Okay. Now our aim is to select the fourth one, right? And do not allow the third one to grow. However, both are going to grow here, because in both the plasmid, both the bacteria contain the plasmid, and in both plasmid, ampicillin resistance gene is functional. Here also ampicillin resistant gene is functional, here also it is functional. So ampicillin resistance gene is functional in both the cases. Therefore, both the bacteria will become resistant to ampicillin. So you will get certain colonies of bacteria here. Okay. Now, if I can take another petri dish, for example, here if I take tetracycline containing medium, a medium which is now containing tetracycline, then what will happen? Think little bit. You will see that the third possibility will be able to grow here, because here the tetracycline gene or resistance gene is active. So the third one will grow, but fourth one, by the process of insertional inactivation which we studied just now, we have introduced the foreign gene within tetracycline medium. So tetracycline medium became tetracycline resistance gene became inactivated. So the fourth one will not be able to grow. As a result, what has happened? While tetracycline, we were interested to get the fourth type, but we could actually get the third type, right? We wanted the fourth type where the selection and of the recombination can be done, but the third one only got selected. That third one was able to grow. Try to understand. Why third one is able to grow? Because third one has the plasmid where tetracycline resistance gene is active. So the bacteria is tetracycline resistant. Fourth one, tetracycline resistant gene is inactive. So the bacteria is susceptible to tetracycline. It is not resistant to tetracycline. In such a situation, what we have to do, this is not there in your book. In details, it is not there. Here in this situation, a little cumbersome process has to be carried out. What we do is, first, we will grow the bacteria in ampicillin containing medium. So now here you have the colonies of bacteria which are both recombinants as well as non-recombinants, both are present here. But only transformants are there, non-transformants are not there. So what we do is we give a particular direction to this particular plate. Let us say north, south, east, west, like this. We do a method which is called as replica plating method. We call it as replica plating because we make a replica, a copy of the first plate. A copy is made. How is it done? You can simply take a, let's say, a kind of measuring beaker. You can just take a beaker. At the back side of the beaker here, you will put a velvet cloth and press this beaker first against this Petri plate. This is the first step. So obviously you will take a beaker large enough whose bottom will be having a diameter almost same to that of the Petri dish. So first step, you will press it against the first plate. At the second step, what you will do is, without changing the direction of the beaker, you will press it against the second plate, keeping the directions of both the plates same. That's why I am giving direction here as north, east, west, south. What will happen is exactly at the same location, some of the bacteria from the first plate will get transferred. Right? Now, when you allow it to grow, some of them will grow and make colonies like this. Some will not be able to grow. Let's say these are not growing. These are growing. Now, since you have kept the directions of both the plates same, you can compare and you can know that from the previous plate, first ampicillin containing plate, this one did not grow, this one did not grow, this one did not grow. So these are the three colonies which did not grow in the second plate which contained tetracycline. Thus, what does it prove? That these are the three colonies which did not grow in tetracycline containing medium. They are the recombinants. Rest are the non-recombinants. So you can now easily isolate these bacteria from these colonies, which are recombinants. This is called as a replica plating method. This is a little cumbersome method, but it is one method which can be carried out. Okay. So anyone has to ask anything in this process, please tell me before we proceed to the next process.
Sir, can you explain it again?
Okay, I will. So basically this process is not tough, but students we have seen usually gets confused in terms of what is resistance, what is susceptibility and all. First of all, you should know resistance means the organism is unable to grow in the presence of something. So here we know that antibiotics are those chemicals which can kill an organism. Mainly we talk about bacteria. Second thing is we talk about genes here which are antibiotic resistance gene. Now these are those genes which are responsible for conferring the property of resistance towards a particular antibiotic. So if a bacteria contain that gene, then that you cannot kill that bacteria with that antibiotic. We have two examples of antibiotic resistant gene here. One is ampicillin resistant gene, other is tetracycline resistance gene. Ampicillin resistance gene is the one which, if it is present in the bacteria, then the bacteria become resistant to ampicillin. Means you cannot kill this bacteria with ampicillin. So when we grow the bacteria in the laboratory, we make certain medium which are called as bacterial growth medium. Now, if your growth medium you put any antibiotic, normally the bacteria, all bacteria will die. But here, when we put suppose ampicillin antibiotic, then those bacteria who have the gene will not survive. Those bacteria who do not have the gene will be able to survive. So out of our 4 possible bacteria, in 2 of them transformation did not happen, which means the plasmid that contains the antibiotic resistant gene, it did not enter inside the host cell. Therefore, that host cell in the first and second case, they are not resistant to antibiotic, and thus they will continue to be susceptible to that antibiotic. Means if the medium contain the antibiotic, then they will not be able to grow. So when in the first place we took ampicillin in the growth media, then all those bacteria which did not have the plasmid, they did not grow because the bacteria does not have antibiotic ampicillin resistant gene, because the gene is present in the plasmid and plasmid did not enter the bacteria, so bacteria does not have the gene. So the first and second type, which are known as non-transformants, where transformation did not happen, they will not survive. The third and fourth type, where transformation did happen and the ampicillin resistant gene is successfully entered along with the plasmid vector and is active, that will be able to grow in the first plate. Now our second step is, from the third and fourth types possibilities, we want to separate the non-recombinants from the recombinants. So we want the recombinants to grow, non-recombinants should not grow. Okay. Now we know that when we did the recombination, we have cut the tetracycline resistant gene, okay, and inserted the foreign gene within the tetracycline resistant gene. As a result, the tetracycline resistant gene became inactive. Now the plasmid, if it enters the bacteria, then the bacteria will not have the property of tetracycline resistance. So it will not be able to grow in the media which contain tetracycline. Right? So what we do here is, we carry out a technique called as replica plating. This is called as a replica plating method, wherein the first plate which contained the growth media and where the bacteria were growing, we make a replica of that plate, copy of that plate. How do we do it? We take a beaker. At the bottom of the beaker, we put a here we will put a paper, right? And we will press this beaker against the first plate so that some of the bacteria from the colonies will be transferred to this velvet paper, and then we will press that beaker into another plate which contains tetracycline media. Now we know that the successful recombinance in which tetracycline resistant gene is inactivated will not be able to grow because the bacteria is no more tetracycline resistant. But by keeping the direction of first and second plate same, and by transferring the bacteria from a location of first plate to exactly same location at the second plate, we can easily compare that which colonies from the first plate, bacterial colonies from the first plate, did not grow in the second plate. Correct? So when we compare this and we go back to the first plate, then we can identify which one did not grow in the second plate. The ones which did not grow in the second plate are the ones where recombination successfully take place. And therefore, from those colonies, you can easily isolate those bacteria which are successful recombinants as well as transformants. These are those colonies which contain recombinants as well as transformants. Okay, this is a very important topic. The phenomena which I told you, wherein the gene is inactivated by inserting a foreign gene inside it, known as insertional inactivation. This comes in the exam. You must understand what exactly is insertional inactivation. The concept is simple. Literal meaning is you inactivate something by inserting something within that gene. So one gene is there which is active. If you insert another gene within that gene, then the original gene will become inactive. That is basically the concept here. Now, in this process, when you have to do this replica plating and all, so the process becomes little cumbersome. So there is another process which can be carried out, which will be easier. Okay. What we do in this process, understand. In this process, the plasmid has different type of marker gene. Let us say this is your plasmid. Here, let us say you have one antibiotic resistant gene, which is ampicillin resistant gene, that is fine. You have another gene. In the previous case, it was tetracycline resistant gene. Here it is a beta galactosidase gene. Okay. Beta galactosidase gene. So it gives you an enzyme called as beta galactosidase. Now what we do is, in a plate, this is a petri dish. This one contains ampicillin medium, as well as it will also contain something called as X-gal. This is a chromogenic molecule. Means it can develop color. Now understand that this beta-galactosidase gene, this gives you an enzyme called as beta-galactosidase enzyme. It will break down this X-gal, which is a galactoside compound, into a blue colored compound. Okay. Now this plate contains ampicillin as well as X-gal. Now when this ampicillin is present, like discussed before, the first and second will not be able to grow. Why? Because in the first and second case, you have not seen transformation. So the bacteria is not resistant to ampicillin. So they will not grow. The other two will grow because other two has ampicillin resistant gene in the plasmid. But this time what we have done is not an antibiotic resistant gene, but a different gene has been inactivated, which is called as beta-galactosidase gene. Now we know that this gene is responsible for breakdown of X-gal into a blue colored compound. So you will find some colonies which will be blue in color. Okay. This is suppose a blue color colony, this is a blue color colony, this is another blue color colony, suppose. And the other colonies you will get their white colored colonies. So blue colored colony means what? Blue colored colony means these are the colonies which contain bacteria, okay, with active beta-gal gene, that is beta-galactosidase gene. These are the colonies which have bacteria with inactive beta-gal gene. Right? What does it mean? If the bacteria contains active beta-galactosidase gene, means there is no recombination, and therefore these are not the one which we are looking for. But if there is inactive beta-gal gene, inactive because there is a successful recombination, these are recombinants. Okay, a successful recombination, the foreign gene got inserted into the your plasmid, okay, and within the beta-gal gene it got inserted, and thus beta-gal gene became inactive. Therefore, the property of this enzyme means the enzyme is not produced at all. If the enzyme is not produced, galactosidase enzyme is not produced. If it is not produced, then you will definitely not get blue colored compound. Correct? So this is an easier process. Why easier process? Because in this case, you can get the result in one step itself. You don't have to do plating or growth of bacteria into two different plates. Okay? Is it understood? Any questions? Please tell me. Both the processes, is it clear? Anyone has any more question? Please tell me. So I hope it is clear for everyone. The selection process. Selection process is a very, very important process. Without doing selection, you cannot get a successful recombinant one or a successful transformant one. So selection is a necessary process when we look at the overall steps one after the other, there you will understand where this selection process take place. Okay, and what will be the significance of this process. This is the one. This diagram explains how the insertional inactivation take place. Okay. Beta galactosidase gene, also called as LACZ gene. Now, this was all about the vectors that can be used for bacteria, wherein the most commonly used vector found is the plasmid vector in case of a bacteria. But sometimes you might need to introduce a gene into a plant cell or maybe an animal cell. Then you will need vectors specifically for plant cells or animal cells. Okay. Now, here comes another discovery which you can call as a revolution itself. Okay. That till now, whenever we saw any plant infected by a bacteria, infection I am talking about, okay, then the bacteria will usually not enter inside the cell. The entry of DNA inside the cell is a property of a virus. Viruses insert the foreign DNA into a cell. Bacteria never do that. While bacteria do not do that, bacteria usually remain in the interstitial space, means in between the cells. Okay? But there was one bacteria which was discovered, known as Agrobacterium tumefaciens, very important. Okay. This bacteria contains a plasmid known as Ti plasmid. And Ti plasmid contain a part of DNA called as tDNA. Okay. This is the Ti plasmid. Right? Has different sequences in it. One of the sequence starting from here, this is the left tDNA border to the right tDNA border. This part of the Ti plasmid is known as tDNA, where T stands for basically transfer. And for the first time, it was discovered that this particular bacteria has the ability to naturally introduce this Ti plasmid into the bacterial cell, where Ti actually stands for tumor inducing. It is a tumor inducing plasmid. So this Ti plasmid, whenever this is the picture of a bacteria. This Agrobacterium tumefaciens, whenever it causes an infection, it causes a disease called as crown gall disease. Okay? These are some of the pictures. You can see in the roots it is caused, in the stems also it is caused. Okay, this is a kind of tumor that forms in the plant. Okay, now this tumor is caused because the bacteria, whenever it infects, it will introduce this Ti plasmid, and this Ti plasmid contained a region known as virulence region. This virulence region is responsible for causing tumor. It causes tumor. Right? Now understand what scientists have done is they have taken the advantage of this particular natural phenomena. They came to know that this bacteria is able to introduce this DNA into the plant cell naturally. This is a natural phenomenon. So we wanted to use this particular property of the bacteria and use the bacteria to introduce the foreign DNA into the plant cells, where it was understood that it is this transfer DNA region which is mainly responsible for the transfer of the plasmid. But the problem was it contained this virulence region which can cause tumor. So a certain part of this virulence region is removed from this plasmid. Okay. The removal of these parts of virulence region, okay, it contains genes which are known as Vir genes. Vir stands for virulence. So these Vir genes, when they are removed, maybe some or entire part is usually not possible, but some part of the virulence gene, when it is removed, then it loses the property of virulence, means it loses the property of causing any type of disease or disorder. Okay? This phenomena by which the Vir genes are removed, this is called as disarming. Remember the word disarming. Disarming means to make the DNA disarmed, means it will lose the property to cause any kind of problem. However, the tDNA is not disturbed. So when you do not disturb the tDNA, the property of transfer of the plasmid into the host cell, host plant cell is still retained. So now this tDNA, Ti plasmid, if it is introduced, if it is allowed, then it will introduce itself, or the bacteria will introduce the Ti plasmid into the host cell, but it will not be able to cause the disorder. Okay? So when we remove the virulence gene here, at this region, a gap is created. So here you can easily introduce your gene of interest. Okay. So you replace, you can use the word replace. You can replace certain Vir genes with your gene of interest. And thus, if you put this Ti plasmid into the bacteria, allow the bacteria to naturally infect the plant cell, they will introduce the Ti plasmid into the plant cell without causing the tumor, but it will introduce your foreign DNA into the plant cell. Is it understood? This is a very, very important technology which has developed and helped a lot in the field of what we call as agricultural biotechnology or plant biotechnology, etc. A similar technique was used for animal cells also. We know about retroviruses. Retroviruses are those viruses which contain RNA genome. Like HIV is the most common and first understood, detailed understood retrovirus. Tobacco mosaic virus is a retrovirus. Our current coronavirus is a retrovirus. So these viruses have a very high infection efficiency, which means it can infect the animal cell with very high efficiency. Okay. So again, scientists wanted to use this property of the retroviruses to introduce foreign gene into animal cells. But the problem is retroviruses are often very, you can say, pathogenic viruses. So similar to the TI plasmid of Agrobacterium tumefaciens, you have to do the disarming of these retroviruses also. And then this disarmed retrovirus can be used as a vector, wherein you can introduce your foreign gene of interest and put it inside the animal cells. According to the experiment, according to the host, you can use these different vectors. This was all about the vectors as such. Okay, so I believe you people have understood about the cloning vectors. Now, second. Now, next thing is competent host cell. So I have discussed it in the beginning that you have two challenges. One is the host cell will usually not take up the foreign DNA so easily. Now, we have discussed about how vector is important for introducing a foreign DNA into a host cell. The logic was that the host cell will not be able to recognize the foreign gene and will not accept the foreign gene. Now, second thing is there are two possibilities. Number one, there may not be any vectors available. Okay, sometimes for a particular host. That is something we need to understand. We said if bacteria is a host, then we'll use plasmid. But it is not necessary that all bacteria will contain plasmid. Right? Similarly, if I talk about animal cells, we said we will use retrovirus. Now, retroviruses or any virus we know are very host specific. So what if you want to introduce a particular gene into a cell for which there are no specific retrovirus? Okay. What if the Agrobacterium tumefaciens is unable to infect a particular plant cell? It's not true that all plant cell will be infected by Agrobacterium tumefaciens. Okay. So although those vectors are available, but the vectors are often specific for specific host. So there may not be sometimes a vector available for a host. Secondly, use of vector is also sometimes a little cumbersome process, a difficult process as such. We often look for some easier process. Okay. Moreover, another important point is, although we said that in case of bacteria, there will be a vector that will be responsible for introducing or carrying the foreign gene into the host. But when we talk about transformation, means entry of the DNA inside the cell, any DNA, whether it is foreign DNA or plasmid DNA. Plasmid is also a DNA only. So transformation of the DNA will not happen so easily unless the bacteria allows the entry. Okay, like in one of my previous class, I told you, I gave you an example, an analogous example, where we said that if someone wants to come to your home who does not know you and whom you do not know, means a complete stranger, definitely you will not allow the entry. So if that person has to come to your house, what he will do most probably, he can take help of someone whom you already know. Okay. So he will come along with that your known person. So that your known person becomes a vector. But now both the person, although they reached your home, to be able to enter inside your home, what is necessary is you should open the door. If you do not open the door, then whether outside it is your known person or unknown foreign person, does not matter, none of them will be able to enter the house. So this opening of the door by bacteria, okay, or such type of host cell allowing the entry of the foreign DNA, for that you need to make the host cells competent. Competent means, in a way, you make them capable of taking up the foreign DNA. So there are a few techniques we will discuss here which are used for making the host cells competent. First one we are talking about is for bacteria. Since DNA is usually hydrophilic in nature, we know that, okay, lipid loving. Therefore, when you look at the plasma membrane of any cell which is made up of lipid, it will go and get stuck to the lipid layer. It will not enter inside the cell. So because of hydrophilic nature, the naked DNA cannot be taken up by the cells. So there are various techniques which are carried out. One common technique that is carried out as given in our syllabus is called as heat shock treatment. In this particular process, heat shock treatment, basically what is done is simple. You take the cell and put it in a temperature of around 42 degrees centigrade. Let us say you take a vial. Here you have the buffer where you have the host cell. Okay. This tube, okay, you will put it into a temperature of 42 degrees centigrade approximately for around, let's say, 10 minutes, 15 minutes approximately. Then immediately you transfer it to a ice cold condition. Okay. So there is a sudden change in the temperature. That change in the temperature is called as heat shock. Understood? And with this, when you also add calcium chloride, this calcium chloride induces some small openings which are transient openings. Transient means what you know? Transient means for a short period of time. Okay, 2-3 seconds, like that. So immediately at that time, if you put here the DNA molecule, the recombinant DNA molecule, that is your plasmid with the foreign gene, that will go inside the cell during that period of time when these transient openings are formed. Immediately these openings will close down. Okay. Secondly, there is another process which I have not mentioned here, that is called as electroporation. It is a similar technique here. What is done is, electro means electric current, poration means pore formation. Here we will take the DNA as well as the host cell in a medium Here you will have the recombinant DNA and the host cell, a mixture, and you will put here two electrodes. Both these electrodes, the two electrodes through which when the current will flow, then a very small amount of voltage, very small, minute voltage is allowed to pass through. That will create what we call here as electric shock. Okay. So heat shock, electric shock, both are responsible for creating transient pores. Heat shock is a better method because in electric shock, many a times the cell destroys. Okay, cell get destroyed. Now for bacteria, this is one technique which can be done so that the DNA will enter inside the cell. Now, disarmed pathogens, we already discussed how Agrobacterium tumefaciens and retroviruses after disarming can be used for transformation. This is one technique which is called as micro-injection, that is done for animal cells. Look at the diagram. Diagram will explain this process very easily. This below you see the pictures of actual instrument. Okay, this is how it will look inside the microscope. First understand, you have a pipette which is holding pipette, a small pipette also called as micropipette. Then it will be responsible for holding the host cell. Okay, and here you have a injection. This is called as a micro injection, very small injection. In this micro injection here, you will take the DNA molecule. This micro injection will pierce through the outer membrane, cell membrane, then nuclear membrane, and it will deposit the DNA into the nucleus. If you recall, there was a similar process you have studied known as ICSI, intracytoplasmic sperm injection. Exactly same process. Difference is there you use the sperm and not DNA. Here it is a DNA molecule. And there injection is done in the cytoplasm. Here injection is done where? In the nucleoplasm, inside the nucleus. Correct? So this is not something, even ICSI has a similar instrumentation. Okay. And under microscope, it will look same like this. But here you can easily make out the needle is going inside the nucleus to deposit the material there. Material is foreign DNA. This is a process which is done for animal cells. If you do not find the retrovirus technique possible for transformation into animal cell, then this can be done. Third is known as gene gun method or biolistic method. This is basically done for the plant cells. For plant cells, it is done. Now, what happens? You see, look at the principle of the instrumentation. You see here, you have the tissue. You can see the tissue is kept at one place. You have the instrumentation in such a way that you have a plunger here. Okay, you can see the diagram. It literally looks like a gun. So this is the plunger of the gun. With the help of helium gas, pressure is produced, and this membrane called as restraining membrane. Okay, this will be ruptured, creating a vibration here. You can see these vibrations are created. Shock wave. This will come and the vibrations will be coming to this particular disc, which is called as a Kapton disc. Here there will be some particles, gold particles. Normally we use some gold particles or tungsten particles like that. Okay, here some gold or tungsten particles, which will be coated with DNA. Gold or tungsten is used because it is inert. That will be coated with DNA molecule, right? So because of the shock wave produced from here, this disk will be moving at a very fast velocity, and it will strike against this platform where the disk will be retained. Disk will not go beyond, but these particles will move through these pores in this membrane, or stopping screen we call it as, and they will collide with the cells here. Because of this high velocity collision, the DNA will pierce through the walls of the cells, that is cell wall, and enter inside the cell. Okay? Basically, it's like how you use any gun, right? So you can say this micro projectile is like the bullet of a gun, but you have some DNA coated gold or tungsten molecules here, while the micro projectile will not go farther. It will be stopped here, the disk will be stopped here. Through small pores in this screen, these DNA molecules with the coated gold or tungsten molecules will move at a high velocity towards the tissue. This is called as a biolistic or gene gun method, which is widely used for plant transformation, plant cell transformation. These processes have both positive, negative effects. Biolistic or gene gun method, if you say, then the process is easy process if we see, but success rate is low. It often leads to destruction of the either DNA or the cells in the tissues. Even when we talk about micro injection, the process theoretically looks like so simple and easy, but it is not so easy. The success rate is very low. Piercing of the cell itself is responsible usually for the death of the cell. When you pierce the cell through the membranes, it often leads to the death of the cells. That's why we said even ICSI is not a process which is so easy. You have to carry out this process, microinjection, multiple times. Okay, if you carry out 100 times, maybe a few times you will become successful. But still, this process is carried out widely because it is easier as such compared to manipulating a retrovirus and all. That is more cumbersome process. So these are all the processes that are carried out for making the host cell competent, means making the cell capable to take up the DNA, or the technology by which the foreign DNA is introduced into the host cell. So that will be all for our today's class. Tomorrow we'll be talking, or next class whenever we meet, we'll be talking about the actual process now using these tools. Okay, so that was all for today.